Hello everyone, welcome to the channel Microbial World. In the previous video, we discussed the initiation and elongation steps in prokaryotic transcription. Today let's learn the final step, which is termination of transcription in prokaryotes. Prokaryotic termination is the process by which transcription of a gene in prokaryotic organisms is halted, which means termination marks the end of transcription. The transcription process will stop. It involves the termination of RNA synthesis and the release of newly synthesized RNA molecule. In other words, we could say that RNA polymerase dissociates from the DNA template and release the newly synthesized RNA transcript. There are two main types of termination mechanisms: Rho-dependent termination and Rho-independent termination. In Rho-dependent termination, termination is mediated by Rho protein, a helicase enzyme. Whereas in Rho-independent termination, the termination is mediated by the RNA transcript itself. It does not need protein factors like Rho protein. Let's discuss more about Rho-independent termination. It is also called intrinsic termination. As we already said, this termination mechanism does not require Rho protein. As the RNA polymerase transcribes a gene, it encounters a specific DNA sequence called the terminator sequence. The terminator sequence contains a palindromic DNA sequence. So what is palindromic DNA sequence? It means the sequence is same when read from left to right on one strand and from right to left on the complementary strand. This palindromic DNA sequence forms a stem loop structure in the RNA transcript due to the base pairing interactions within the palindromic sequence. It is also called hairpin structure. Downstream the stem loop structure, there is a stretch of uracil residues in the RNA transcript. This uracil-rich region further destabilizes the RNA-DNA hybrid and promotes the dissociation of RNA polymerase from the DNA template. The stem loop structure is followed by a GC-rich region. GC-rich region stabilizes the stem loop structure and enhances the efficiency of termination. Now let's see how Rho-independent termination occurs. Terminator sequence. As we already said, as the RNA polymerase transcribes a gene, it will eventually encounter a specific DNA sequence known as the terminator sequence. The terminator sequence contains two elements: a palindromic DNA sequence that can form a stem loop or hairpin structure in the RNA transcript, and a stretch of uracil residues located downstream of the stem loop structure. Hairpin structure formation. The RNA transcript will form a stem loop or hairpin structure. And how is this hairpin structure formed? It is formed due to the complementary base pairing interactions within the palindromic sequence. Pausing of RNA polymerase. Now the formation of this hairpin structure will cause the RNA polymerase to pause as it transcribes the terminator sequence. Weak RNA-DNA interaction. As the RNA polymerase encounters the hairpin structure, the stability of RNA-DNA hybrid decreases. The weak interaction between the RNA transcript and the DNA template facilitates the dissociation of RNA polymerase from the DNA template. Release of RNA transcript. RNA polymerase along with the newly synthesized RNA strand dissociates from the DNA template. Termination of transcription. So with the release of the RNA transcript, the transcription of gene is terminated. The RNA polymerase is now free to initiate transcription at another gene. Rho-dependent termination. It is a termination mechanism in prokaryotes that involves Rho protein. Rho protein is an ATP-dependent RNA helicase protein that plays a crucial role in termination. It binds to the newly synthesized RNA molecule and moves along it in 5' to 3' direction. Termination signal recognition. As the RNA polymerase transcribes a gene, it will encounter a termination signal. This signal consists of a stretch of uracil-rich sequence followed by a GC-rich region that forms a hairpin structure in the RNA. Rho binding. When the RNA polymerase pause at the termination signal, the Rho protein binds to the exposed RNA transcript. And where does the Rho protein bind? Rho protein binds at the Rho utilization site. Rho helicase activity. The Rho protein utilizes energy from ATP hydrolysis to move along the RNA transcript towards the RNA polymerase that is paused at the termination signal. RNA-DNA dissociation. As the Rho protein catches up to the RNA polymerase, its helicase activity will unwind the RNA-DNA hybrid. Once the RNA-DNA hybrid is unwound, the RNA transcript is released from the RNA polymerase. The Rho protein will continue to move along the RNA and finally dissociate from it. With the release of the RNA transcript, the transcription of gene is terminated. I hope that the videos on prokaryotic transcription was helpful for everyone. Do subscribe the channel for more informative videos. Thank you.